This is Dr. Momcilovic's Bio 182 Lecture 4.1 on the origins of life. In this lecture, we're going to discuss some of the theories on how life on Earth started. We're going to begin by looking at the history of the Earth according to geologic time and the fossil record. So the Earth formed about 4.6 billion years ago. Animal life that we would actually be able to recognize as animal life, started showing up about 1 billion years ago. So there's quite a bit of time between the formation of the Earth and the time when living organisms were showing up on the planet. So when we look at these different eons, eras, periods, and epochs, one thing that is important to look at or to notice is that things that end in zoic, like Paleozoic, zoic means animals. So it's at the end of the Precambrian when we start seeing animals and life showing up on Earth. So the Cambrian is where we started first seeing animals showing up on the planet. The Cambrian period is when we started seeing most of the phyla that are alive today evolve. Following that, we had the Ordovician, the Silurian, and the Devonian periods, which were known as the ages of the fishes. Following that, we had the age of the dinosaurs, which is the Permian, the Triassic, the Jurassic, and Cretaceous periods. The Tertiary period is where we started seeing mammals begin to radiate and flourish after the dinosaurs had been wiped out. The Quaternary is where humans appeared. About 2.4 million years ago, the genus Homo seems to have evolved. And therefore, and then if we look at the Pleistocene epoch within the Quaternary period, that would have been when we would have seen cavemen and early humans. The Holocene epoch is the time period that we are living in now. That's modern man. So each period or epoch is distinguished by a change, either in land masses, types of organisms, or an asteroid impact. The one between the Cretaceous and Tertiary, or between the Mesozoic and Cenozoic eras, would be where we had the large asteroid hit that wiped out a large chunk of the dinosaurs on the planet. All of the dinosaurs did not go anywhere. That's why we have things like birds. Birds are evolved from small dinosaurs. However, the changes between the periods and epochs is defined by a large change in landmass or organism or something that created an enormous extinction event. When we're talking about the history of our planet, there are several changes that have occurred through time that have impacted the life that we see on the planet. The first one would be changes in the atmosphere. Early Earth had an atmosphere that had no oxygen, and over time, oxygen accumulated, which allowed other types of organisms to survive and flourish, and changed the metabolism that living organisms used to be able to survive on the planet. Through geologic time, climate has also changed quite a bit. There's been periods of hot and cold, and those have fluctuated. Back in the Proterozoic, which was about 2.5 million years ago, we started getting periods of glaciation. And so one of the arguments that people who don't believe in climate change will make is that the climate on the planet has changed and fluctuated throughout the history of the Earth. And therefore, any change in climate is normal and should be happening. Most climate change deniers don't deny that there is change happening. They simply believe that the change is normal. However, we know that the current climate change is happening very fast. It's happening quite a bit faster than it has in the past. And it seems to be human induced, which is a new phenomenon. And therefore, there is normally large fluctuations in the climate of the Earth. However, it's slow enough and gradual enough that organisms have time to adapt or they go extinct. What's currently happening on our planet is that rate of climate change is so quick that organisms are unable to adapt to that climate change, and therefore they are more and more becoming threatened and endangered and are going to go extinct in the near future. The last geologic change that we see occurring on the planet involves land masses. So if you look at the video that's going on down in the bottom left-hand corner of your screen, it's showing the change in how the land masses on the planet have been joined and connected and separated over the history of the Earth. And that happens through a process called plate tectonics. So plate tectonics is where we have the surface of the Earth, which is covered in tectonic plates, which are floating solid rocks on top of liquid magma. And there are convection currents going on in the magma that move those plates around on the surface of the Earth. And that movement changes things about our atmosphere, about the temperature of the planet, about the location and the temperatures of the continents as we know them. So I do encourage you to go back into the lecture slides and watch this occurring. It's very, very interesting to watch how the plates on the Earth have moved to get to the current location. And those plates are constantly moving. And so hundreds of millions of years from now, the continents will not be arranged in the same way that they currently are. One of the most important things that we use to be able to learn more about early life on Earth are fossils. A fossil is any preserved remnant or impression of an organism that lived in the past. Common fossils include bones, shells, feces, footprints, or burrows. Fossils can be an actual part of an organism that used to be alive, such as a bone or a shell that has accumulated calcium over time and hardened into rock. Or it can be something like a footprint that through sort of the same processes becomes part of that rock structure. You can also have imprints of things such as plants, or where you have the cells of plants be replaced by calcium carbonate and therefore create a fossil over time. Using fossils, we can attempt to reconstruct what ancient ecosystems looked like based on the types of organisms that we find fossilized in the same areas. Our fossil record is not complete because most things don't actually get fossilized. It is very, very rare for any individual to be fossilized at a point in time. It requires very, very specific environmental and sedimentary parameters, as well as the right type of structure that the organism is made of to be able to be fossilized. It's a lot easier to fossilize things that are made of shells and bones than it is to fossilize something that is made of less rigid structure. So fossilized cells or fossilized fungi, fossilized amoebas, things like that, things that don't have a very hard structure are hard to find fossils of. Fossils of bones, shells, even footprints are much easier to find than are other types of fossils. And on top of that, those fossils have to be discovered and then described, which is another task on top of the originals. So it's quite rare that things get fossilized, and then the chances of us finding and being able to extract and describe those fossils is limited. So we don't have a complete fossil record from the beginning of life on Earth to the current day, but we do have a lot of fossils that sort of fill in the blanks of how life on Earth started and how it has evolved to where it is today. One of the most important things about fossils when we find them is to know how old they are. In order to help us fill in those blanks of the fossil record, we need to know how old this fossil is and when in time this type of organism was alive. There are two sort of different categories of methods that we can use to date the age of fossils. The first one is called relative dating, which is where old fossils are buried in deeper layers and younger fossils would be found closer to the surface of the Earth. This is how scientists would have dated fossils 100 or so years ago, where they did not have access or the technology had not been developed yet for us to date fossils. The issue with relative dating would be that it assumes that rock layers are never intermixing and moving around. And we know that that now is not true. And therefore, relative dating has been determined to not be that reliable of a way of dating the age of fossils. The other category of fossil dating is called absolute dating. This is where we use radiometric approaches, meaning that we measure the amounts of radioactive isotopes that are present in the fossils. Anything that is from 0 to 40,000 years old, we can use carbon dating. Carbon dating is the type of dating that most people are familiar with, but it's only useful for fossil specimens that are 40,000 years old or younger. And that's because of the way that radioactive carbon decays. So carbon-14 would be the element that we are looking at when we're doing carbon dating. And that type of carbon has a half-life of 5,700 years. By the time we get to about 40,000 years back in history, the reliability of using carbon for that dating process, it becomes unreliable. There's so little now to work with it, it's hard to estimate how much is left. For anything more than 40,000 years old, we use what we call potassium-argon dating. In potassium-argon dating, we are actually dating the age of lava. And so intermixed among all these rock layers, we can find hardened lava in the fossil record. And so the way that this works is that we have to find lava above where we found the fossil, meaning that it would probably be younger, and lava below that fossil, meaning it would be older. And so from that, we are able to sort of make bookends of the time period in which that fossil would have been alive. And so when scientists give the age of a fossil specimen, they usually will say, oh, it's 1 to 2 million years old. And the reason for that is because anything over 40,000 years old, we have to use potassium-argon dating. And for potassium-argon dating, we are testing lava. And so we have to test at what time period the lava in the fossil record before that fossil occurred is, and then one after it. And so we can sort of find the range of time that that fossilized organism would have lived, but pinpointing the specific point in our history where that organism was alive is a little bit more difficult until someone comes up with a new method for dating fossils. We can also use molecular evidence to look at the history of life. DNA is a very important tool for establishing the relationship between organisms. We usually think of using DNA to look at the relationship between living organisms or individuals of the same species or even people. However, DNA has mutations, as we've discussed, and those mutations occur at predictable rates. So we can look at the DNA of different organisms and look at the mutations that have occurred in comparing those genomes between the two. And from that, we can sort of look back in time at when their nearest divergent date was. So when we're thinking about humans and chimpanzees, based on the amount of mutations in their genomes, we can estimate that the most recent common ancestor between humans and chimpanzees was about 5 to 6 million years ago. And we do that based on the mutation rate, which is generally very constant through time, and the number of mutations that differ between humans and chimpanzees, and therefore we can estimate or hypothesize the point in time at which humans and chimpanzees diverged from each other. In case you guys aren't familiar with the phylogeny, which is this branching diagram that you see both, there's two separate ones at the bottom of this slide. A phylogeny is just a hypothesized relationship between organisms. And anywhere that you see a sort of a branching point is a common ancestor between two species or groups or whatever it is that you're looking at. So in some cases, we may not have yet found the fossil remains of predicted ancestors, but a DNA analysis can help with clearing up the relationships. So we can do that for humans, we can do it for any other mammal, we can do it for insects. We just look at the differences between their DNA as well as different mutations, and we can estimate how long ago they shared a common ancestor. So for evolutionary biologists, one of the things that is studied extensively is evolutionary transitions or the acquisition of new traits. So if we're talking about a marsupial, them gaining a pouch in which to carry their babies. If we're talking about reptiles and amphibians and birds, the ability to have a hard-shelled egg or the evolution of wings, things like that. But before we get to that, we need to explain two of the most important transitions. So before we can even discuss the transitions between life, the first thing we need to do is discuss abiogenesis, which is the origin of life from no life, and endosymbiosis, which is the evolution from prokaryotes to eukaryotes, which will be our next lecture. So abiogenesis is the creation of life from no life. So anytime that you see a at the beginning of a word, it means not. So in this case, we're saying that from nothing, life was formed. So the Earth is 4.6 billion years old. But how did the first living organisms arise from the nothing that was in existence on Earth at that time? From research in several different fields, we have information that tells us about the atmosphere of the early Earth. We know that that atmosphere had no free oxygen, but it had a lot of methane, ammonia, hydrogen sulfide, and hydrogen. our modern atmosphere is made up of about 78 or so percent nitrogen, 21% oxygen, and the remaining 1% or so is other gases, including argon, CO2, neon, helium, that kind of stuff. And so we know what's in our atmosphere currently and what keeps things alive. But considering the atmosphere of the early Earth, how did life begin with none of the environmental gases and things like that that we would need for life as we know it to occur? One of the most widely accepted hypotheses for how life came from nothing was created by Stanley Miller and Harold Urey in the 1950s. And this idea is what we call the primordial soup hypothesis. And it's the idea that chemicals that are found on early Earth spontaneously reacted to create molecules that assembled into the first living cells. To test their hypothesis, Miller and Urey set up a very simple but brilliant experiment where they had a set of interconnected tubes and vials where they had in one essentially a replication of the gases that we think were available in the primitive atmosphere, with electrodes to create lightning. We think lightning happened quite often during the formation of our current Earth. And then some water, which would have been the ocean, as well as a heat source. And when they ran this experiment again and again, that started with nothing but water and gases and some electricity, which would have been created by lightning. So they were replicating the Earth's early atmosphere, as well as possibly the environmental conditions where most of the surface of the Earth was covered in water. There were simple but important molecules that were generated. They made from these gases and water amino acids, which are the building blocks of proteins, purines and pyrimidines, which should look familiar to you guys because they are the building blocks of RNA and DNA. So Miller and Urey's experiment was groundbreaking because they were able to test a hypothesis on the conditions of Earth's early atmosphere and what would happen if those conditions were occurring. Since the 1950s, there's been a lot more research into the early Earth's atmosphere, and we've added a few more things that we think would have been in existence. And this same experiment has been rerun many, many more times with additional gases and other things that may have been present. And the more of those things that we add in, we are able to create more and more sort of building blocks of proteins and things like that. And so we continually find evidence using the same methodology that this primordial soup idea, where we just had an ocean and a lot of gases and elements and a lot of energy from lightning, would have possibly been enough to create the building blocks of life, which is profound and exciting. So we don't just need amino acids and the building blocks of DNA and RNA for life. We need a few other things. We need cell membranes, metabolism, and a genome. Cell membranes can actually be created spontaneously when phospholipids are put into a polar solution such as water. They will orient themselves into shapes that begin to look like cell membranes. And interestingly, they will create a phospholipid bilayer. And when you think about the structure of a cell membrane and trying to move things across the membrane and those electrical potentials you guys would have talked about in Bio 181, you start to see that there's a potential there to start making some type of reaction or metabolism. So having some phospholipids available could have created the earliest type of cell membrane. A genome seems to be a little bit trickier to create out of nothing. In the past, we've always assumed that RNA formed first and then DNA evolved later. We think this for several reasons, including that RNA can self-replicate itself and catalyze proteins from that. New data, very, very new data, as of earlier this year in 2020, would have actually shown that that proteins were possibly able to be the precursors even to RNA. So proteins that were able to self-replicate may have come before even RNA, and that's very, very new data. So to talk a little bit more about this, I want you guys to go back into the lecture notes and watch this video on the last slide for these lecture slides and learn a little bit more about building protocells and the origin of cells.